Hello, we're going to talk about chapter 5 in this video, and that covers the plasma membrane. Now, I know we already talked about the plasma membrane when we talked about cells and when we talked about lipids, but this week we are going to get into the plasma membrane and see what it does for the cell. This is a really nice micrograph picture taken with a TEM microscope, transition electron, right? So we're looking at the inside of a cell, and it's actually two cells that we're looking at. So this here is one cell, and then this here is another cell, and then this dark part is the intracellular space, so that empty space between two adjacent cells. Now, if you look really closely, you can see the cell membrane of each cell, and it is a double layer, just like we've been studying. So it's really interesting to look at the cell on this microscopic layer, or microscopic TEM microscope, and to see that it really is a double layer. Okay, so we're going to cover each section like we normally do. So 5.1, we'll talk about the fluid mosaic structure that the cell membrane is. We'll describe phospholipids, proteins, and carbs. I know we already talked about them, but we're going to talk about what they do for the cell membrane. And then we'll look at membrane fluidity. So the plasma membrane has a lot of functions, which we know, defining the outer border of the cell. It also defines the outer border of organelles inside the cell. It manages what enters and exits the cell, so it's able to keep that interior environment of the cell at a certain range of set a set range than the exterior part of the cell. It also receives external signals from elsewhere in the body, especially if you're a multicellular organism. You need to receive signals so that your cells can work together and initiate a response. And they also adhere to neighboring cells. So we've talked about the junctions that cells have. If you remember them, gap junction, desmosomes, and tight junctions are those junctions that animal cells have. Plasmodesmata are the ones that plant cells have. So you can simply not breathe without the cell membrane. The cell membrane is responsible for taking in O2 and using it in the electron transport chain. So this is, the cell membrane is called the fluid mosaic model. It looks something like this, and it is made up of more than just phospholipids. So it has a lot of proteins, it has carbohydrates, it has things called glycolipids, glycoproteins, it has cholesterol in it, and all of these give that mosaic part to the fluid mosaic model. Now, the fluid part comes from the ability of these phospholipids to move around. So they're not stuck in one place. They are able to move and rearrange themselves and trade places. And so it's not a hard structure, it's fluid. And that allows cells to move and absorb shock and stress and change shapes to their environment. So phospholipids, just going to cover them again really quick. Phospholipids are almost like a triglyceride, but instead of that third fatty acid tail, it has a phosphate group there. So that gives it this hydrophilic characteristic. Now the tails are still going to have that hydrophobic characteristic, so they are not going to like water, and they will be situated inside of the membrane, away from water. So, just to recap, a phospholipid has two fatty acid tails. Those fatty acid tails can be saturated with hydrogens, like this one, or unsaturated with hydrogens, like this one, and typically there is a double bond somewhere that makes a kink. There is also that glycerol molecule here in the center, and then that phosphate group up here on the head region. Okay. So phospholipids, again, are the major component of the plasma membrane. Now, phospholipids can be arranged into a layer, a single layer, but more than likely you will see them arranged into a double layer or a bilayer. Now, in the lab, the phospholipids will arrange themselves this way on their own just because of their chemical characteristics. The heads, again, they like water, they're hydrophilic, so they will orient themselves outward. And then the tails, they do not. So they're going to hide in the center of the layers here in the bilayer. So if we take a look at this animation, you will see these areas of phospholipids orient themselves in a certain way. The green portion are the tails, and then the pink, red, blue portion are the phospholipid head, which contains the red phosphate group, and then the blue glycerol molecule. So over time, you'll see them organize themselves with the tails facing inward, heads facing outward, and eventually it will organize itself into a sheet, which is pretty cool. Okay. All right, so proteins are another major component of the plasma membrane. So there are a lot of different types of proteins, and they serve a lot of different types of functions depending upon their makeup. So there are, first off, there's integral proteins, which go all the way through a bilayer, and then there are peripheral proteins that are only on one side of the layer. So, integral proteins typically are going to act like channels. They will allow certain things in and out. Peripheral proteins are usually used to bind to something. Usually it's binding to the cytoskeleton. It might be binding to another cell if it's on the outside. It could also carry a carbohydrate on it. So this glycoprotein, so there's the protein, there's the carbohydrate part, that's going to be oriented outward. And so usually the cell uses that as a recognition molecule to say, hey, that's our self, or that's a cell that I know, it's okay. Or it will recognize this as maybe a pathogen and the cell will be destroyed. So the way that these integral proteins are organized are by going through the membrane, and there are certain ways in which we can do that. So it can pass through the membrane one time, it can pass through a few times, or it can pass through a bunch of times, and that's a bigger protein if it passes through a bunch of times. So if you remember, the bilayer, again, it has different chemical properties, right? On the exterior of the layer, it's hydrophilic, which means it likes water. On the interior of the membrane, it's hydrophobic, which means it does not like water. And so proteins, if we think back to the chapter where we covered proteins, chapter 3, proteins are made up of amino acids, right? And those amino acids have different chemical properties. So remember, the primary structure of a protein is the links of all of these amino acids together in certain orders. So if we need a protein that is going through the membrane, we need to have certain amino acids in order. So a non-polar amino acid is going to be perfect to be situated inside of the membrane. So if this is one long protein strand, this will be a repeat of of non-polar amino acids here. And then on the exterior, you'll probably have polar amino acids, or something that would like to be on the outside. So just keep that in mind when we talk about proteins. So again, proteins are very specific on what they do. The primary, secondary, tertiary, quaternary structure, very specific on the protein function. If that changes, the protein changes. And any number, a combination of these is going to make a different protein, right? So integral proteins are very integral for the plasma membrane. So let's move on to carbohydrates. So I already mentioned that carbohydrates are located on the exterior of some of these proteins. They are also located on the exterior of some of the lipids. So take a close look at this one here. Instead of a phospholipid head, it's been replaced with a carbohydrate. And glycolipids are just used in recognition. They are used for binding, cell-to-cell recognition, and also attachment. So carbohydrates mostly present on the exterior of the membrane. So they act again as a receptor or a recognition site. So this is the HIV virus. Notice it has a lot of recognition sites on the exterior of it. This is a cell, a human cell here. So those recognition sites will meet up with recognition sites on a cell, and that actually recognizes HIV as itself, and it lets HIV into the cell. So it thinks it's okay, that it's part of your body, and then that is allowed to enter the cell, and that's how HIV infects your body. So let's talk about membrane fluidity. So we talked about the mosaic with the proteins, the lipids, the carbohydrates. So the fluidity portion of the fluid mosaic model is represented or is affected by mostly the phospholipid type. So remember, we have saturated fatty acids, which are straight, and then unsaturated fatty acids, which are bent. So a membrane made up of mostly saturated fatty acids is going to be viscous, and so it's going to be more rigid, and it's not going to move a lot. There's not a lot of things that can enter and exit. It's not healthy for the cell to have a lot of saturated fatty acids. So a fluid membrane looks more like this. There's more unsaturated fatty acid tails, and so you can see there's more spacing in between phospholipid heads because of those tails. And so now the membrane can bend a little bit more, it has a little bit more give, and that's better for the cell. Now things can enter and exit a little bit more easily. Of course, you don't want it to be too fluid where the cell will break apart, but just enough unsaturated fatty acids is perfect. So another thing is cholesterol. So cholesterol is a little molecule that gets embedded in the tail region. So remember, cholesterol is a lipid. It does not like water. It's hydrophobic. So it's going to be in the center of the phospholipid bilayer. Now, cholesterol adds a little bit of a space between phospholipid heads, and that allows the membrane to be more fluid. So this is really good for animals that are subjected to cold temperatures, like cold water fish. They will typically have more cholesterol in them so that their cells don't harden up with the cold temperatures, and it also helps cells become not too fluid when it's hot. It provides a type of like anchoring part of the cell. So temperatures, when cold temperatures are acting on the cell membrane, it's going to compress the phospholipids together a little bit more, and that'll make the phospholipid bilayer more rigid and not fluid. So phospholipid type, fatty acid tail type, cholesterol presence, and then temperature are three things that will change the fluidity of the membrane. Now there's a really nice recap video that you can watch here. Alright, so here's a poll question. Which characteristic of a phospholipid contributes to the fluidity of the membrane? Which characteristic of a phospholipid? So that is going to be letter D. Double bonds in the fatty acid tail are going to contribute to the fluidity of the membrane. So the head, cholesterol is not part of a phospholipid. A saturated fatty acid tail is not good. That's going to take away from the fluidity. So double bonds, which create an unsaturated fatty acid, will contribute. Here's a nice worksheet that I have that goes over the plasma membrane. You can color it. It tells you which color to color, and then it also tells you to write the letter that goes with the structure or the function below. And here's a nice video that recaps recaps cell membrane is by Bozeman Science. Okay, so that's 5.1, the components and structure of the plasma membrane. So hopefully now you know what fluid mosaic means. Hopefully you can describe the parts that make up the mosaic part, and you can describe why the fluid part is important and what changes the fluidity. So 5.2, we are going to talk about passive transport. We will explain why and how passive transport occurs. We'll hopefully understand the osmosis and diffusion process and define tonicity and its relevance to passive transport. Okay, so passive transport happens on its own. Typically, the plasma membrane itself is selective. It's selectively permeable, so it's only going to allow certain things in and out of it. So the cytosol solution will be different from the exterior or extracellular solution. So the interior of the cell is going to look different from the exterior of the cell, and that's due to its selectively permeable nature. So transport across the cell membrane can be passive, which typically happens on its own, and it requires no energy. And then there's also an active transport that can occur, but that requires energy, just like the name implies. So first we will talk about passive transport, and there's another related term, facilitated transport, and also osmosis and diffusion. We'll talk about those first. Okay, so selectively permeable. That means that only small nonpolar molecules can enter a phospholipid bilayer. So things like oxygen, carbon dioxide, and lipid hormones. So they can go through a membrane. So anything that's large and has a charge to it, negative or positive, it's not going to go through the membrane. So passive transport, the simplest type is diffusion. So diffusion occurs when there's a substance of high concentration in one area and it's low, or there's really no concentration on one side. So diffusion is going to occur down the concentration gradient or with the concentration gradient, and that will even out the distribution of whatever substance that is. So as long as this substance that we're looking at here is small and it's not charged, it will move through the membrane. So here's a nice GIF from Amoeba Sisters showing you how passive diffusion, passive transport can occur. So it goes this way. So we'll talk next about what facilitated transport is, but first we'll talk about how diffusion can be affected, how diffusion rates can be affected. So a lot of factors that affect diffusion rates. One is going to be the concentration gradient, how great the concentration gradient is. If it's a large gradient, then diffusion is going to happen faster. Now, if there's a large mass of molecules, meaning that the molecule is really big, the molecule is not going to move fast at all. However, if the molecule is small, then that molecule will move quicker and there will be a quicker diffusion rate. Temperature. So high temperatures, hot temperatures, diffusion will happen faster. Low temperatures go slower. Solvent density. So, just like the mass of the molecules, if the solvent weighs a lot or is very dense, then it's going to diffuse slower. Okay. Surface area. So if we have a larger surface area for our cell, then diffusion will happen quicker. If our cell does not have a big surface area, it has more volume, then diffusion will happen slower because there's just not enough space on the cell membrane for those molecules to go through. So distance travel. So this is more for like a hormone. So if the distance that the hormone needs to travel is very far across your body, then it's going to take a really long time for that diffusion to happen. However, if the distance is very, very close, then diffusion will happen fast. Pressure, so in some cells, like in the kidney, blood pressure can affect the diffusion rate. And then solubility, if nonpolar materials are present, they diffuse faster. So meaning that they can slip through the membrane faster. Okay, poll question. The principal force driving movement in diffusion is what? That's going to be letter C. So the concentration gradient is the #1 driving movement of diffusion. Okay, so we looked at this GIF earlier, and you probably noticed this one over here, and there's something extra on this diagram. So this one is titled Facilitated Diffusion, and it's facilitated by an integral protein. So that integral protein provides a channel for the substance to move through. So typically things that need a channel are going to be things that can't do this, right? So they're going to be things that are ions, meaning they have a charge, plus or minus charge, or they're polar and they can't get through that middle layer. They're just blocked. So there are two types of transport proteins, channel proteins and carrier proteins. Notice that the diffusion is still going to occur from high concentration to low concentration. The only difference is that there's a protein involved. So no energy involved yet. Okay, so transport proteins, some can be opened all the time. Some of them can be gated. So like when there's a signal that's received, it will activate the protein and it will open up. So there's some specific examples. Aquaporins are specific types of integral proteins and they let water through. So muscle cells also have gated channels allowing for muscle contraction when they're opened. So they're not open all the time, which is a good thing because then you would have a muscle contraction all the time. So they need to be gated and they need to have a signal in order to open. So facilitated diffusion can also use carrier proteins. So carrier proteins let a molecule in and they spit it out the other side. It's not just a channel. They can't let another molecule in until that one molecule moves through. One example is a glucose transport protein. So glucose is a fairly large molecule. It's not going to move through the membrane on its own. It needs help. It needs a protein. And so this one is carried by a carrier protein. Okay, so osmosis is another way that we can do diffusion. Osmosis is just referring to water moving across a membrane. So water likes to move to dilute a substance. So if we take a look at this GIF here, the green circles are sugar, the blue circles are water. So at the start of the GIF, you can notice that there's a lot of sugar molecules here, and there remains sugar molecules here only on this side by the end of the GIF. So, since the sugar molecules can't move through the membrane, then water is going to move through the membrane to dilute the sugar on this side. So by the end of the GIF, you should be able to pause and count the number of water molecules, and it should be roughly equal on both sides. So just keep in mind that water dilutes the solute. So really sugary on this side, there's no sugar on this side. Water is going to move towards the sugar to dilute it. In other terms, water is going to move down the concentration gradient. So water is in high concentration here. Water is in low concentration here because of the sugar. So it's going to move down the concentration gradient. Cystic fibrosis occurs because osmosis is not occurring properly. So cystic fibrosis is a disease of the lungs and airways where the body does not release water like it should. And so it's actually due to a faulty chlorine pump or chlorine carrier protein is what I want to say. So with a healthy individual who does not have cystic fibrosis, chlorine is pumped out and that actually attracts water. And so water moves out of the cell to dilute the chlorine. In people who have cystic fibrosis, that chlorine stays inside of the cell, so water stays in there to dilute the chlorine that's present, and so it doesn't move out. So whenever you get phlegm in your throat, instead of being able to clear it normally and have it slide through your airway, it just sticks to your cell and it clogs up your airways instead. Okay, so more about osmosis. So osmosis causes something called tonicity, and there's different types of tonicity. There's hypertonic, isotonic, and hypotonic. So they're going to describe the cell in relation to the exterior environment. So in a hypertonic solution, the water is let me show you on the next slide. Okay, in a hypertonic solution, the water is going to leave the cell. So that means that the solution that the cells are in has more solutes in it. So water is going to move out of the cell to dilute the solutes that are outside in the solution. So on the other hand, if these cells were placed in pure water where there's no solute, water is going to move into the cell to dilute the solutes that are inside of the cell. And most of the time, that'll cause a burst of the cells because they take on too much water. And then in the middle is isotonic, and that's the best one. So that's where we have homeostasis and equilibrium. So if we take a look at this GIF here that explains it a little bit better, hypotonic solution, there's a lot of water outside of the cell and it moves into the cell to dilute the solutes inside and the cell gets bigger. Isotonic, there's equal exchange. Hypertonic, the water inside of the cell moves outward. So usually it's a salty or a sugary solution on the exterior that's causing water to move out of the cell and causing it to shrivel up. Okay, so here's an activity that I like to do with my students. I have them draw the following pictures on their own paper, and they're going to indicate with an arrow which way water will move. I also have them label each as hypo, hyper, or isotonic. And then I ask them which one has low osmolarity and which one has high. So if you want to do this, one thing I'd point out is that each of these only tell you the water concentration. So everything should equal 100%. So the solution outside should equal 100%. The solution inside should equal 100. So this is fine, 100% water, but there's 2% missing. So I would write 2% solute here. And then 2% solute, 2% solute, and then 2% solute, and 5% solute. So that might help you answer the question of hypo, hyper, iso, and the questions about osmolarity. So if you did the activity, what you should have is that water is going to move out of this cell, water is going to be exchanged evenly in this cell, and water is going to move in for that cell. For the first cell, the solution that the cell is sitting in, so this, is going to have high osmolarity, and then this solution will be low osmolarity. So good job if you got those correct. So real world problem, a doctor injects a patient with what the doctor thinks is an isotonic solution. So they think they're injecting them a solution that is equal to the solution that's already inside their body. So the patient ends up dying, and the autopsy reveals that many red blood cells have been destroyed. So do you think the doctor injected an isotonic solution? So the doctor did not inject an isotonic solution if the blood cells were destroyed. So what do you think it was? Well, it was actually a hypotonic solution, which caused the cells to take up too much water too fast, and it caused them to burst and be destroyed. Okay, so plants and animals can do osmoregulation. Plants particularly are easy to observe doing osmoregulation because they have a cell wall. So organisms with cell walls prefer hypotonic solutions because it creates a nice push or turgor pressure against the cell wall. So things like plants, fungi, bacteria, protists, they don't have bones, so they need some type of structure. Well, providing this pressure against the cell wall makes that organism rigid, and it makes that organism withstand harm, right? So an hypotonic solution where the vacuole has a lot of water in it causes the plasma membrane to push against the cell wall. So in an isotonic solution, the plasma membrane you can see is not really super tight against the cell wall. And then in a hypertonic, maybe it hasn't rained in a while, the cell membrane pulls away from the cell wall, and that causes the plant to wilt. So why can't you water a plant with salt water? Well, it causes a hypertonic environment where it draws water out of the cell instead of water being taken up by the cell. So turgor pressure is observed in plants that have not been watered versus plants that have been watered. So this plant, not having been watered in quite a while, was looking more like a hypertonic solution. And the plant that has been watered, or this is the same plant just before and after being watered, has nice turgor pressure, and you can see the leaves are not shriveled up anymore. Organisms like protists can do osmoregulation as well as plants, and so this paramecia lives in fresh water. So that means that the interior portion of the cell is going to be in higher osmolarity, which means it has more solutes inside. And so it has to continually pump water out of the cell, because if this has a salty environment in the cell and this is fresh water, water is going to continually move into the cell by osmosis. That's just how it happens. So this paramecia has contractile vacuoles that collect excess water and pushes it out of the cell. So marine invertebrates that live in salt water have internal salt concentrations that match their environment. They don't really osmoregulate, but fish, they do osmoregulate. So fish that live in salt water excrete a lot of diluted urine to get rid of excess salt. So they drink a lot of water, but they don't urinate as much. Most of their urine is really, really, really salty, so that it gets all that salt out of their body. So here's a nice diagram showing that. So here's our saltwater fish, drinks a lot of water, but the only thing that they're really excreting is a really, really concentrated urine. So a freshwater fish, not going to drink much water because water enters through the body on its own, but it's going to pee a lot. So it's going to have a lot of urine, but it's keeping that salt inside of its body. Okay. So this was supposed to be a video that was showing a paramecia, but it's not on here. But paramecia do live in fresh water. So the tonicity of the environment would be a hypotonicity, right? Because water is constantly moving into the paramecium. If the paramecium was put into an opposite type of an environment, the paramecium would probably shrivel up because it would lose water. And then what if it was put into an extremely low osmolarity environment? Well, same thing. It would shrivel up. Okay, poll question. In which situation would passive transport not use a transport protein for entry into a cell? So take a look at the answers. So we're talking about passive transport, but we don't need a protein. So that should be D, because oxygen is very small and it's not charged and it can move directly through the membrane. So good job if you got that one right. The ion cannot move directly through the membrane. Glucose is too large, and water is also, it's not too large, but it's polar. So it has a slight charge and it can't move through the membrane. All right, so that's 5.2, passive transport. So hopefully now you can explain how and why passive transport occurs. It occurs down the concentration gradient. Hopefully you can understand what osmosis is and the diffusion process, and you can describe tonicity and passive transport. Okay, so 5.3, active transport. We'll talk about how transport can occur with energy, and we're going to understand electrochemical gradients. Okay, active transport is used by something that can't move through the membrane on its own, and it typically occurs against the concentration gradient or against an electrochemical gradient. So when I say against a concentration gradient, it's going to go from a low concentration to a high, which is the reverse of what we've been talking about. So it's like trying to take a person from the street and push them onto an already packed bus. So you have to use a lot of energy to do that. Electrochemical gradients can be set up where one side of the membrane is very positively charged and the other side of the membrane doesn't have a charge or it's negatively charged, and we're trying to push more positively charged towards the positive side. So these two things are happening against the gradient, and so we're going to have to use energy to make it happen because it just doesn't happen on its own. It's against physics. Energy is always required for active transport, so active meaning energy. And there are two types of active transport. There's primary, where ATP provides the energy, and then there's secondary, where an electrochemical gradient provides the energy. Now, I do want to say one thing before I move on, that active transport, it usually has to occur all the time, just because normal transport, passive transport, is occurring against active transport. So remember, diffusion is going to occur on its own over time, and the only way to combat that is that active transport acts in the opposite direction. So electrochemical gradients can be set up in this manner where a net charge is accumulated on either side of the cell. So it's not a neutral charge. One side can be positive, the other can be negative. So because ions move in and out with active transport, that causes that change to occur. So here is a nice GIF showing active transport. So notice high concentration here, low concentration here, but we're pushing the molecules from low to high. So that's going to require energy. Typically, sugar molecules are going to be used or are going to be utilizing active transport because we need the glucose in order to make energy. So those will continually be brought into the cell. So things that are always used by the cell are typically brought in that way. They're always going to be used. So they're always going to be actively transported. So there's a few types of carrier proteins for active transport. There's a uniporter, a symporter, and an antiporter. So a uniporter, pretty straightforward, just carries one molecule or ion. A symporter carries two different types of molecules and ions, and then an antiporter carries two different types, but in opposite directions. So primary active transport, remember this one requires ATP. And primary active transport, a great example is the sodium-potassium pump. It moves 3 sodium out and then 2 potassium in. So if we're moving 3 sodium out and then 2 potassium in, what type of porter system, what type of carrier protein are we talking about here? Well, it would be an antiporter because we have different molecules going in different directions. So this creates, this is actually an electrogenic pump. So what do you think that means? Well, it's going to create a different charge. So let's take a closer look at this. If we have 3 sodium, 3 positively charged sodium move out and then 2 positively charged potassium move in, since we're moving 3 out and 2 in, we're going to have a slightly more positive charge on this side of the membrane because we just have that one more charge every time we move these ions. So secondary active transport does not use ATP, and this uses the electrochemical gradient to its advantage, or sometimes it can use another molecule to its advantage. So amino acids and glucose enter the cell this way. Here's glucose molecules, here's glucose molecules, here's sodium molecules. So glucose hitches a ride with the sodium and enters the cell that way. So what type of cell is this, or what type of carrier protein is this? It's going to be a what? A symporter, right? Because it's carrying 2 different molecules in the same direction. So both molecules have to be present in order for this to occur. Oh, and one more thing. So we're still moving against the concentration gradient with this. Notice the glucose molecules, there aren't any out here, there's a lot in here, even though that's the way that they're traveling. So poll question, how does the sodium-potassium pump make the interior of the cell negatively charged, even though both ions are positive? So because the potassium, we're only moving 2 potassium versus the 3 sodium, that creates a difference in charges across the membrane. So that's going to be C, because we're expelling more cations than we're taking in. So good job if you got that one right. Here's another poll question. Active transport must function continuously because why? So D is the correct answer. Diffusion is constantly moving solids in the opposite direction. So diffusion is going to happen on its own all the time. So active transport combats that and moves the solids in the direction that they need to go. Not necessarily want to go, but the direction that they need to go in. All right, so that's 5.3, that's active transport. So hopefully now you can describe what an electrochemical gradient is and distinguish between primary active transport and secondary active transport. All right, so bulk transport is our last little section here. We're going to talk about endocytosis and we will talk about exocytosis. So bulk transport is just referring to the import or export of a lot of molecules and particles, or very large molecules and particles. So bulk transport is a type of active transport. Energy is required for bulk transport, and it's also called endocytosis if we're taking things in, or exocytosis if we're putting things out of the cell. So 3 types of endocytosis, bulk transport, are phagocytosis, pinocytosis, and then receptor-mediated endocytosis. So we're going to take a look at these here in a sec. Phagocytosis is also termed cellular eating. Phago is food, cytosis, cell. So the cell membrane is going to surround a particle and create like an invagination into the cell membrane, and then that cell membrane will come together and pinch it off, and now you have a vacuole. So typically that is going to be transported to a lysosome, and that lysosome will meet up with the vacuole. They will merge, and the lysosome has digestive enzymes in it, and that digests whatever molecule or a large particle or group of molecules that are here. So if we take a look at this video, a macrophage is going
Neutrophils are white blood cells.
this macrophage is going to chase and digest a bacteria cell. So notice how the white blood cell is chasing it, and eventually the bacteria loses and the white blood cell wins, and it takes that whole bacteria in by phagocytosis. Just like that. So it happens very quickly. Pinocytosis is called cellular drinking. I like to think Pinot like Pinot Noir, Pinot Grigio, like the wines, and that's how I remember that term. The cell is going to create a vacuole with liquid or water and dissolved materials or minerals in it, and it creates this vesicle. So again, a lysosome will meet up with the vesicle, digestive enzymes will be released, and the material inside of the vesicle will be digested and broken down into smaller usable parts. Receptor-mediated endocytosis is another type of energy-intensive bulk transport. So this uses receptors to recognize different molecules. So notice there's 2 different types of molecules here. Only the star-shaped ones are the ones that the cell is interested in. And so it has specific receptors for those. And once those things are recognized by those receptors, or they attach to those receptors, then the cell releases these things called clathrin, and it creates a vesicle, and it's a very intensive process, which you can learn more about in the textbook, but we won't cover all the steps here. Just know that receptor-mediated endocytosis uses receptors, so it's a little bit more specialized. So those were endocytosis. There's 3 different types of endocytosis. Exocytosis is just the release of materials to the exterior environment of the cell. Typically, it's large things, or it's a lot of materials at one time. So let's think back to the endomembrane system. Remember, endomembrane system, it has the Golgi apparatus, and from the Golgi apparatus, those vesicles go to the cell membrane. So this is what happens when those vesicles go to the cell membrane. They meet up with the cell membrane, and then those materials are released. So it could be waste products, it could be proteins that need to go elsewhere, it could be pretty much anything. Okay, poll question: which of the following organelles relies on exocytosis to complete its function? It's really D, right? So all of the above. So exocytosis needs to occur from the ER. So even though it's not the cell membrane, it still has a membrane. So exocytosis will occur from the endoplasmic reticulum. That endoplasmic reticulum goes to the Golgi. The Golgi uses endocytosis to take it in, and then it packages it, uses exocytosis to take it out, and then that's a vacuole, and then that vacuole will go to the cell membrane to complete its function, or to another organelle. Okay, and that's bulk transport. So hopefully now you can describe endo and exocytosis, and then the different types of endocytosis. And that is it. So hopefully now you have learned about diffusion and the cell membrane. Hopefully now you can describe what I mean by the fluid mosaic model, and you can describe how things get across that membrane, including water. So water is a really important one. Make sure you take a second look at those tonicities.